Good day, everyone. So the story presented here is about the competitive advantage that we provide to our customers and collaborators by delivering performing, easy to adopt, and cost-effective solution for Germline workflows, focusing on library prep for whole genome and whole exome sequencing, and somatic NGS workflow with an emphasis on the extraction of total nucleic acid from FAP tissue samples and DNA shearing for comprehensive genomic profiling. Covaris is a standard of excellence for DNA shearing. In the past 20 years, we kept increasing our knowledge and understanding, trying to understand what are the pain points that we see out there in the field. And what we know today, what we see, is that compared to enzymatic-based fragmentation method, the bias fragmentation is still a problem, and that can be seen through the different GC content, especially at the beginning of the read, the duplication rate, especially from PCR-free library prep, and the variable insert size. Some of the causes for these failures or challenges are variable processes that are due to the different purification kit and different DNA sample sources that they should be equivalent, but actually they are not when you use enzymatic fragmentation, and also because of batch-to-batch reproducibility that is still like a issue and a concern in the field. With this variable process, ultimately, if we consider like a whole exome sequencing, they may drive to a suboptimal hybrid capture that translates into a poor exome enrichment, and this translates into long over-sequencing or a poor usage of the flow cell. In addition to that, the implementation, there are, I will show references and real case stories, that this would lead to long optimization time when enzymatic fragmentation is used, and sometimes even the need to split pipelines depending on what sample sources are used, or what exome panel is used as well. So these are the three pillars that are really the foundation on our focus and dedication to the genomic community. We look at the ease of use and adoption of our solution. We aim to continuously improve performance for next generation sequencing application, and we are looking on how to improve the economics of the lab, especially in a period where cheaper and faster is becoming important requirement especially for clinical application. The claims presented here are backed up by data and real case study with customers that I will show you in later slides. So DNA fragmentation using Covaris ultrasonication is a standard of compatibility. Boasting over more than 20 different library prep options. Here are just the main ones that are used in the field. And they cater really to a different need for sequencing. In addition, the library prep process can be integrated in a fully automated modular solution or manual, delivering the need for different batches and different throughput. In looking at the compatibility across sequencer, there are some recent publications from Element Bioscience where our fragmentation has been featured. And I would like to cite especially this paper that they show a higher performance of whole genome sequencing compared to Illumina. And the trick that they use there is they use longer fragments. Their technology allows to sequence long fragments without having the bias of preferably amplifying shorter fragments. And the fragment size used for this particular, reported in this paper, was 1250 base pair, which can be a game changer for whole genome. So the study indirectly demonstrated the value of tunable DNA fragmentation that is provided by Covaris, where we can fragment between 150 base pair to 4 kb, while enzymatic fragmentation is really limited to few base pair. I haven't seen data that goes beyond the 200 to 500/700 base pair. Now, this is a project that is ongoing with Zach Herbert, that is the director of Dana-Farber Genomic Course Lab from Dana-Farber Institute. Our objective is to uncover improved chemistry and condition that will keep improving our superior performance and cost advantage compared to enzymatic fragmentation. So in this project specifically, one of the preliminary data is the GC bias that is presented at the first 10 read of a 150 times 2. And while we notice that for Covaris, the representation of the bases is what is supposed to be, based on the percent from a human genome, that on average is like 41% for GC, the enzymatic fragmentation is generating some base insertion at the very beginning, and this represents between 6 to 8% of your total read, with impact, with what we believe is an impact not only on the alignment, but also ultimately at the calling of the variants that are required in order to have a very robust analysis. There is a reference here where they evaluated 5 different library prep using enzyme, and all of them, they show bias like this. Another data point to prove the enzymatic fragmentation methods are generating bias fragment is the duplication rate from PCR-free libraries. They can be up to 18%. What does it mean is that the enzyme, preferably, especially for repetitive sequences within the DNA, tends to cleave the DNA in certain position. These are read as duplicate, and they get discarded, while they may have important information from downstream application. Here we present the data shared by Quest at a lab seminar in collaboration with Covaris. In their comparison of mechanical fragmentation against enzymatic, we show a high degree of reproducibility of fragmentation at around 200 base pair, which is the optimal insert size for exome, all exome sequencing, while enzymatic fragmentation show a lot of outliers. When we deconvolute this and cluster them, divided by sample type, we see a high degree of repeatability within the sample, but also reproducibility across the different sample. How does it impact the economics? First, based on this paper, they did an elegant study where they proved the exome efficiency, the capture of the exomes based on different fragment size, and you see that the window in order to have an optimized capture is actually pretty limited. So what does that mean is that the longer are the fragment, or also shorter, the less efficient is the exome capture, and then when it goes into the sequencing, because of the preferential amplification or shorter fragment, your overall exome coverage goes down, and they saw an improvement of 3x compared to their former enzymatic library prep. Now, this is another real-world scenario. This is a collaboration we did with Radboud University in the Netherlands, Hamilton, and Twist for their exome capture. Over 339 samples, we got a 98.2% pass rate on the coverage for the exome, with an average of 109x. And this is excellent considering that the process is fully automated. The R230, our sonicator, was installed on a Hamilton Star Plus, and the full library prep is completely hands-off. This here is more like a teaser that we are trying to understand better. The data from the literature is scattered, so it's very difficult to estimate what the QC failure is when using enzymatic fragmentation. We kind of estimate it is around 11%, especially considering all these variables, sample types, and capture for exome. And I would really like to have further discussion with the people in this room or beyond this room, if we can improve your workflow, if this data is resonating with you.